Katie. I'm a senior at Yuma Catholic High School and I'm coming here for undergrad.
Hi, my name is Amaya. I am a current junior at the University of Arizona studying biochemistry, and this is the Dr. Compilation lab.
Our lab focuses on researching the effects of menopause, its cardiovascular implications, and how we can influence it by using the microbiome and probiotics.
Many different focuses are worked on in the lab, but all of them influence and overlap one another. The experiments are related to researching the mouse intestinal tract, probiotics, cells, and menopause. Inducing cardiovascular issues is used to determine the collision of menopause and heart failure. Fecal matter is collected to determine the ability of probiotics to reduce the damage from a heart attack in menopause by measuring beta-gus. More are conducted to study the intestinal stem cells derived from menopausal mice and the implications for screening cardioprotective probiotic species. While we cannot conduct a huge amount of research, we are given multiple opportunities to observe and learn from what they are conducting in the lab.
We participate in journal club where we share summaries of academic journals and how it relates to what's happening in the lab. It creates a strong foundation for our peers and gives a concise overview for those wanting to develop a more comprehensive understanding. We constantly ask questions to stimulate the growth of new ideas, which encourages a deeper understanding for the researchers too.
Since the data collection and experiments are ongoing, their data has not been analyzed and there is not a straightforward answer to what the results are.
Some questions we have from our lab are, can treatments through diet be used to aid menopausal symptoms in mice? And how old do wax-embedded samples have to be in order to have the most optimal conditions to slice them in the microtome?
So my questions are, since inflammation is the cause of many different types of conditions, can we use the microbiome to be able to treat different types of conditions in the brain as an alternative route to the blood-brain barrier? Another one is, how would this experiment work related to women having one ovary versus two, or those who experience other ovarian issues before menopause?
Hello, I'm Jay Nabeda, and this past year I graduated from Southland Catholic High School, and in the fall I'll be attending Baylor University. This summer I had the privilege of working with Dr. Shirazi in his toxicology lab, where their lab question is, what is the most effective decontamination method to decontaminate the skin from a toxin? To contribute to that, my research project this year was, does a technical decontamination method prove to be more effective than a water wash decontamination method? And so now I'm going to show you inside of my lab. In order to answer the question for my research project, I conducted an experiment that included the following items. This 3D printed foam stand to take pictures throughout the process. This contaminant solution that we made with 10 milliliters of oil-based food coloring and 100 milliliters of olive oil. In my experiment, it consisted of two methods. Both methods consisted of painting the contaminant on a 3x3 area, blow drying for 30 seconds, and then the first method, you rinse it with water for 30 seconds. Dry everywhere but the contaminated area, and then take pictures again. The second method consisted of the same thing up until the blow drying, but instead of water, you'd use a microfiber towel, wipe it off for 30 seconds. Get a wet wipe it off for 30 seconds, and then use the microfiber towel again for 30 seconds. Then take pictures on 1x and 10x. In our experiment, in order to observe the results, we took pictures at 1x and 10x. In the slide, you can see pictures of the first method, which is a water wash, before, during, and after. In this picture, you can see the before, during, and after contamination of method 2. In order to gain an unbiased, quantitative result for our experiment, we showed 10 people each trial of each method of each subject and asked them to choose which method seemed to decontaminate the skin better. And so as a result, the technical decontamination method was superior to the conventional water wash. Thank you guys for letting me talk to you guys about my summer project and for letting me show you around my lab.
Hi, my name is Liana Tang. I'm an incoming junior attending South Point Catholic High School here in Tucson, Arizona. This summer, I have worked in Dr. Shen and Dr. Gertner's Wound Healing Regeneration and Foreign Body Response Lab. My lab's main research question is, how can we perfect the wound healing process? My lab wants to answer this question because wound healing is a complex and poorly understood process with many faults. It's important that we answer this question because the health effects from wound healing process can be harmful and even deadly. For example, take the case of a heart transplant. Even in a sterile environment, a patient may still suffer from the foreign body response and reject the transplant. Even if they don't reject the transplant, patients still run the risk of developing conditions from the scar tissue created in the wound healing process. Therefore, my lab's goal is to perfect the wound healing process. The big world issue that we are trying to solve is the creation of an agent that perfects the wound healing process and the worldwide distribution of it. To answer this question, there are 4 main experiments. There's a breast cancer experiment, incisional wounding experiment, a foreign body response experiment, and a fibrosis experiment. In these experiments, I mostly do rat surgeries. However, this summer I've learned how to section and how to run complicated procedures, such as frozen section IF protocol. These projects are all long-term projects, and so far there have been no results that show significance. However, this school year I plan on coming in to help out and hopefully find conclusive results. My 3 final questions that came up during my lab work is, how do failure and ignorance work hand in hand? Why is it important that we accept and utilize our ignorance? And what is the best way to simplify a complicated and often overwhelming health issue? Thank you so much. My 3 final questions that came up during my lab work are, how do failure and ignorance work hand in hand? Why is it important that we accept and utilize our ignorance? And what is the best way to simplify a complicated and often overwhelming health issue? Thank you.
Hello everyone, my name is Scott French and I'm an undergraduate researcher for Dr. Craig Weinkauf's lab. I'm currently going into my senior year studying neuroscience and cognitive science. Dr. Weinkauf's lab focuses on how vascular risk factors contribute to Alzheimer's disease and dementia. Specifically, our lab's main question is, how does vascular disease contribute to Alzheimer's disease risk over time? Our lab thinks it's important to explore the question of whether vascular risk factors increase risk for dementia over time, because these risk factors are often modifiable through lifestyle changes such as implementing exercise or dieting, or clinical treatment such as surgery or medication. Our lab's specific focus is on how carotid disease, which is characterized by plaque buildup in the carotid arteries, contributes to dementia risk over time. We try to answer this question through our longitudinal study, which incorporates a plethora of testing, including extensive MRI scans, transcranial Doppler to monitor the intracranial vessels in real time, blood collection to quantify biomarkers of inflammation, vascular disease, and neurodegeneration, as well as other tests that quantify systemic vascular burden. As you can see, our study collects an abundance of information from these participants to quantify their cognitive, vascular disease, and neurodegenerative status. However, our lab could not be successful in answering this question without the help of a large team of hardworking professionals who are dedicated to answering this question as well. I'll give you a brief tour of our lab and give you a chance to meet a few of our members. This is our wet lab space. This is where we analyze all of our blood-based biomarkers and process tissue specimens. Now I'd just like to quickly introduce the members of lab who helped make our study successful. First, Suman and Zara, who are responsible for data analysis, Franchelle, Chris, and Emma, who are research coordinators and are responsible for data collection and patient visits. I'd like to introduce Haley as well, who is a PhD student working for our lab, as well as our lab's leadership, Juan Arias, who's our lab manager and senior scientist, as well as Dr. Craig Weinkauf, who's our principal investigator. My work in lab is focused on quantifying the blood-based biomarkers of inflammation, vascular disease, and neurodegeneration. I do this through multiple methods, including using commercially available sandwich ELISA kits, as well as using Luminex assays, which are bead-based and allow you to analyze multiple proteins at once. So far, I've found a few biomarkers that seem to change with asymptomatic carotid disease severity. PDGFR-beta, a biomarker of blood-brain barrier injury, VCAM1, a biomarker of endothelial activation, and YKL40, a biomarker of neuroinflammation, seem to all increase with severe carotid disease, whereas BDNF, a biomarker of neuron growth and survival, seems to decrease in severe carotid disease. We found no changes with CRP, a biomarker of systemic inflammation. Although this data is exciting, there are a few questions that remain unanswered, such as, how do these biomarkers relate to brain changes observed on MRI, and how do these biomarkers relate to cognitive changes within these patients? These questions are extremely important for determining the clinical utility of these biomarkers in the context of neurodegeneration.
Hello, my name is Lucas Wally. I am a rising senior at Basis Tucson North, and I had the privilege to work with Dr. Mitchell Bartlett throughout this summer. Our lab focuses on answering the question, how can focused ultrasound be used as a treatment to treat various conditions and diseases? I primarily focused on using focused ultrasound as a method to treat Parkinson's disease this summer. One of my questions after receiving my project question is, what is alpha-synuclein and GFAP? What I learned from researching and asking my mentor is alpha-synuclein and GFAP are basically biomarkers that can be used to track astrocytes and see if there's interactions between them. And when staining for these interactions, we would like to understand what is happening between them. And this also brought up another question I had. How will we stain for these biomarkers? Another task that I was given was making a protocol for immunohistochemistry, also referred to as IHC. Through this, I learned how to communicate my findings through research I did through scientific papers. One of the main things we did this summer also consisted of doing other projects such as Western blotting. Western blotting is a technique used to separate proteins by their size through gel electrophoresis. Throughout the summer, I was exposed to multiple protein assays and learning how to perform and understand the meaning of them.
Hello, my name is Ryan Mayer, and I am an incoming senior at Florida Atlantic University High School. This summer, Dr. Mitchell Bartlett was also my mentor, and my research project has focused on the role of focused ultrasound in treating neuropathic pain. My first question upon receiving my research project was, how do you assess neuropathic pain? To this, I researched a technique called the von Frey filament test, in which, of course, my next question was, how do you conduct and set up the von Frey test? So, to conduct the test, you will take a set of these filaments, in which the filaments have different fibers on the end of them. The fibers are set up as such where you will place the rat on a wire mesh and then poke the filament through on their foot until a response is evoked. You will start at the lowest force fiber and then move your way up until a response is evoked, upon which you will conduct the up-down method. Researched and have set up a behavior room to perform many other analyses, including the forced swim test, the 8-arm radial maze, the hot plate test, and much more. Throughout the summer, I also did polymerase chain reaction to amplify a DNA sample and analyze it by running it through an agarose gel and using gel electrophoresis to find its size. I'm grateful that throughout the summer I was able to experience a wide variety of techniques, from protein assays to PCR to rat behavior and surgical technique.
Hello, my name is Ravana Abragash, and I'm going into my senior year at Basis Tucson North, and I'm currently 16 years old. Throughout the summer, my lab mentor has been Dr. Weinkoff, and our lab focuses on cardiovascular disease and Alzheimer's research. Our lab's main research question is, what is the impact of carotid disease on cognitive impairment and Alzheimer's?
This is the wet lab where Scott does a lot of hands-on work and Lily, Mohammed, and Haley do data analysis. This is the dry lab, and this is where my desk is and where everyone else's little office areas are. And this is primarily where people do data analysis. This is the lab I work with.
My lab wants to find a connection between carotid disease and Alzheimer's, as it could provide a new pathway for preventing dementia. This is because carotid disease can easily be slowed or reduced through lifestyle changes, medication, and surgical procedures. In the lab, Dr. Weinkoff and his lab members use advanced imaging and neurocognitive testing to see which Alzheimer's specific changes are associated with carotid disease. Then they want to understand how carotid disease leads to those changes, and finally they hope to identify which carotid disease treatments are effective at improving or preventing those outcomes. In the lab, I'm conducting research based on the results our patients get on the neurocognitive test that we give to them, and I'm analyzing those results to see if carotid disease affects neurocognition. More specifically, I'm differentiating between fluid and crystallized intelligence and seeing which one carotid disease affects more. The first of my research was to divide our neurocognitive tests into tests that test for fluid intelligence and into tests that test for crystallized intelligence. After doing so, I took those tests and I compared the scores that patients with carotid disease got on them and patients without carotid disease got on them, and saw if there was a significant difference between the two. Out of the fluid intelligence test, the ones with a significant difference between the carotid disease patient scores and the non-carotid disease patient scores were the MoCA attention test, the trail making test A, and the trail making test B, the Benson delayed recall test, and the verbal fluency F and L. Out of the crystallized intelligence test, the ones with a significant difference were the WAIS coding test, the WAIS symbol search test, the MoCA delayed recall test, and the category fluency vegetable test. The test I used to determine if there was a significant difference between the scores for carotid patients and non-carotid patients was the t-test. These results show that carotid disease seems to affect crystallized intelligence and fluid intelligence almost equally. Some questions that came up during my research and that I'm curious about are, why those tests? Why were those the only tests out of the fluid and crystallized categories that actually had an effect and were significantly different between carotid patients and non-carotid patients? The next question I have is, how can I compare which intelligence is more affected? One thing we struggled with was trying to figure out how we can find out which intelligence is more affected, if it's fluid intelligence or crystallized intelligence, because now we have the scores and we know what tests are affected from each category, but we're not sure what statistical analysis we should do to effectively compare the fluid and crystallized directly to each other and see which one is actually more affected. The last question I had was, are there any flaws in my method of analysis? Are there any better statistical analyses that I could do that would be more valid or give me better answers? All in all, that's what I've done throughout the summer, and I've had a great time with my lab, and I really hope I can come back and do SIMI again, because this was a great experience, and I just want to thank you all for your time. Have a good day.
Hello, my name is Parker Harris. I work at the Unilab with my mentor and PI, Dr. Russell Whitty.
This is our research lab, and this is my PI, Dr. Russell Whitty. We are a medical imaging lab, and our big question is, how can we better image the electrical signals of the human body?
Our lab works to develop acousto-electric imaging. In medicine, there is a great need for imaging of electrical activity within the heart and the brain that is on the millisecond scale and on the millimeter scale. Current imaging modalities are either too slow and bulky, like MRI, which measures blood oxygenation and is on the minute scale, or do not have the spatial resolution required, like EEG, which can measure on the millisecond scale, but its spatial resolution is only around 1 centimeter. Our lab uses ultrasound for both spatial and temporal resolution, which means that it could get to the millisecond, but also millimeter scale. To achieve this, our lab is combining EEG and ultrasound to generate and measure the acousto-electric effect. Those measurements can then be turned into images of electrical currents within the brain. This required our lab to develop a system that can synchronize both EEG and ultrasound. The experiments we run using this system mostly involve using phantoms that we created to measure electrical data and then compare that data to other prevalent modalities. This summer, I helped develop a way to compare our AE system to other modalities. I did this by integrating a hand task to the EEG, which is also compatible with fMRI and other technologies. This hand task will activate the hand area of the primary motor cortex. This activation can then be measured by the AE system, fMRI, and other technologies. The data we get from those scans can be compared because the task will remain the same throughout all of the scans. This will allow us to verify how accurate our scans on the AE system are.
Hi there, my name is Lance Perez. I'm an undergraduate student majoring in electrical engineering here at the University of Arizona. During the summer break, I'm participating in the Summer Institute Medical Ignorance Program, and I've been working in Dr. Russell Whitty's lab.
Dr. Whitty's lab, referred to as UNL, Experimental Ultrasound and Neural Imaging Lab that is, focuses on using sound as a non-invasive and robust medical imaging method. Our lab seeks to answer the question of how we can use hybrid imaging modalities to more accurately diagnose patients and improve treatment decision-making. Vibrational potentials are created when a pressure wave, like an ultrasound pulse, creates a temporary dipole in the ion surrounding, in our case, an electrode. This in turn temporarily creates an ionization electrical potential observed as voltage, which can be read by the electrode, thus giving us a vibrational potential signal. Vibrational potentials can be used in the imaging of electrodes in the human brain. For a variety of neurological disorders like seizures, deep brain stimulation is a common treatment used. After a neurologist determines exactly where the electrode arrays need to be placed, a procedure is performed. It is important for the patient's health and recovery that these arrays are placed in the precise location they were intended for. This verification can be done during or after surgery with a variety of imaging modalities, but most commonly computed tomography or CT. However, these machines can be bulky and expose patients to ionizing radiation, which must be avoided. Ultrasound vibrational potential imaging offers a modality that is low cost, safe to use, and can be used during or after a procedure. For example, by placing an ultrasound transducer against the patient's skull, ultrasound can be pulsed to the skull in order to oscillate ions surrounding the electrodes in the array. This creates a voltage potential that can be measured from the electrodes themselves. Last summer, I was able to collect vibrational potentials in a simple saline tank setup. However, the question this summer was, is it possible to record ultrasound vibrational potentials with a thick skull in between the ultrasound and the electrodes? And will these potentials have comparable amplitude to vibrational potentials when the electrodes were in a saline tank? The experiment to answer this question was as follows. We had 8 needle electrodes encased in an agarose and saline gel inside a skull cap. We had the skull cap set up such that the ultrasound probe could be coupled with the skull with ultrasound gel. In photos shown, you can see gold clip electrodes covered with tape placed on the surface of the skull. These were used to record a different signal than ultrasound vibrational potentials. The needle electrodes were the ones recording voltages when we pulsed the electrodes with an acoustic wave from the ultrasound transducer, which had a center frequency of 0.6 megahertz. As of the making of this video, I have not been able to look thoroughly through the data collected to fully answer my summer research question.
Hello, my name is Emilio Armenta. I am going into my junior year at Yuma Catholic High School. My lab mentor is Dr. Whitty. We focus on photoacoustic imaging and acousto-electric imaging. Our lab's main research question is how can we push the limits of non-invasive energy to image structures inside the body. Here we have Dr. Whitty's photoacoustic imaging lab. My goal in this lab is to use these 2 dyes, India ink and nigrosin, to make fake skin that could mimic melanin. We use this in different experiments. The last thing I want to show you guys is the lab's photoacoustic imaging system. This system works by using a laser that comes through this cable to heat up the skin. That causes the skin to expand and contract, and we detect that because it makes waves. Those waves can get detected by your ultrasound transponder, which is here, and with that we can make an image.
In my time in SIMI, I have come to understand a lot of things, especially about my lab and imaging. My lab is called UNL. UNL stands for Experimental Ultrasound and Neural Imaging Lab. UNL is pushing forward non-invasive imaging. Non-invasive imaging and procedures are needed and preferred because they're safer, cheaper, and faster than invasive procedures. A well-known invasive procedure would be something like an open heart surgery. Also, in my time in SIMI, I have come up with many questions because SIMI encourages questions and also because of the SIMI logs. But I have come up with many questions, and 3 questions that I think about the most are: what imaging techniques can be used to detect cancer and its development early? How can we holistically treat patients when they have to go through more difficult experiences, not just in imaging, but the medical field as a whole? There are a lot of difficult experiences for both doctors and patients alike. And my final question, which connects most to my project, is: how can we make better skin and tissue phantoms? This is important to my project because my project has been based on making phantoms that mimic melanin.
Hi, my name is Sophia Buchanan. I'm an incoming junior at the University of Arizona studying physiology and medical sciences, as well as nutrition with a minor in Spanish, and hopes to go to PA school in the future. My lab mentors are Dr. Marlise Whitty and Dr. Bartlett, and this summer I studied how the lymphatic system plays a role in Alzheimer's disease. The main question that I've been looking into this summer and that I will continue to look into during the academic year is: how can manual massage of the cervical lymphatics decrease amyloid beta plaque and consequently positively impact behavior in Alzheimer's disease? The Alzheimer's disease models utilized are the 5XFAD models, which express 5 human AD-linked transgenic modifications, and this usually appears very aggressively in the mice. So AD progresses really quickly and you'll see symptoms as soon as 4 months of age. Based on a pilot study that was already conducted, we expect the Alzheimer's disease behavior to improve over time. After 2 months of manual lymphatic drainage, we will be performing behavior analysis, which includes testing for nest building, the novel object recognition test, as well as the rotarod test and the open field test, among other behavioral tests. In addition to learning more about what I'll be working on during the school year, I was also able to learn techniques such as immunohistochemistry, Western blotting, and BCA assays. I was also able to watch surgeries such as pharmacokinetic surgery, and we also did a common peroneal nerve injury model on a rat. My first question is: how does systemic inflammation or chronic inflammation affect the degradation of neurons and therefore the progression of neurodegenerative disease? My second question is: how does gut health and gut microbiome affect neurodegenerative disease? My third question is: how can one promote the health of astrocytes and best support their role in maintaining the blood-brain barrier? Overall, I'm extremely grateful for everything that I learned this summer, and I'm excited to continue my research in this lab during the school year.
My name is Kamal Mode. I attend Flowing Walls High School as a junior. With my lab mentor, Dr. Terry Matsunaga, I've been researching the use of microbubbles in ultrasound imaging. As our central question asks, how can we find a cheaper alternative to MRI and CAT scans through ultrasound?
The lab space.
Our lab is full of equipment, including a scale, a hot plate, a vial mixer, and a makeshift pressurizer. I worked alongside my 2 lab mates, Carson and Susan, for a portion of the program.
Research.
So why, you may ask? Well, CAT scans and MRI scans are incredibly expensive, making it inaccessible to those without a financial advantage. Thus, alternatives must be found, and thanks to Terry Matsunaga and his colleagues, it has. Introducing microbubbles, micron-sized particles that are able to reflect ultrasound waves in order to generate an image. The microbubbles are injected into the bloodstream at nanoparticle size. They are then activated within through either heat or acoustic energy so that they may be bubbles again. Rather than through magnetism or X-rays, we are able to generate images through acoustic reflection alone. Unfortunately, these bubbles are far from perfect. They continue to collapse under high acoustic energy or high temperatures, resulting in our continuous research years after its conception. During my time here, research has involved using various compounds in order to reinforce the shell of the microbubble. Compounds include hyaluronic acid, PLGA, and canola oil. Experimentation involved adding the new compounds into the mixing process in order to reinforce or coat the microbubble shell. While my time here hasn't resulted in any major breakthroughs, I'd still say I learned a lot. A lot about motivation, curiosity, and the overall scope of research. At the end of it all, I'm faced with 3 major questions. 1. What other polymers can we use to reinforce the microbubble shell? 2. What is the result of using multiple compounds in order to reinforce the microbubble shell? 3. What other phase contrast agents can we use for ultrasound? Thank you.
Hi guys, my name is Olivia Rencher. I will be a junior at Marquette this year, studying biomedical sciences on the pre-med track. Over the past month and a half, I have been working with Maria Gracia Morpinos and Katarina Fisher in their efforts to study wound healing and tissue regeneration. The main question my lab wants to answer this summer is whether cultivating a population of myeloid and monocyte cells in a DB mice or DB patient will advance the results of tissue regeneration and wound healing following the day of injury. To answer this question, we decided to use a mouse model for its anatomical and clinical relevance. We first wound the animals, making a small incision on their back. Unlike humans, muscle surrounding a mice's wound would contract, snapping their skin shut and healing the wound in a matter of minutes to hours. To inhibit this biological response, our lab has decided to use a silicone ring. Once that incision is made, we will suture a silicone ring around the incision to hold the skin in place, ensuring that muscle will not contract and heal that wound rapidly. In doing so, we can then study their tissue regeneration and the cells and the upregulation of genes that is going on while that tissue is repairing, and it's more so mimicking the tissue regeneration response of a human so that we can study it for wound healing treatments and future advancements in the clinical field. Days after the incision is made and the silicone ring is set in place, we will then take samples from their skin, bone marrow, spleen, blood, and study that and study those cells and run flow and basically analyze them to advance our research. My main task this summer has definitely been to suture the animals and wound them on surgery days in preparation for sample extraction. Diabetic patients are in a chronic state of inflammation, putting them at a higher risk for a variety of health concerns. With our research, we hope to recover a treatment that will aid diabetics in their wound healing and tissue recovery. I have learned so much this summer, and I only know it will help me in my future endeavors.
My name is Akash Kumar. I go to Basis Ora Valley, and I'm going to be a senior.
Hi, my name is Victor Singster Piet. I am currently an incoming senior here at the University of Arizona, studying physiology and medical sciences.
Hi, my name is Laimani Salazar. I go to Douglas High School and I am a current rising senior.
My lab mentor is Dr. Pires.
My mentor is Christina Hoyer Kimura.
My current lab mentor is Dr. Josiane Fernandez de Silva, as well as Dr. Paulo Pires, working in the physiology department.
So here in the Pires lab, we focus mostly on how cerebral blood flow can affect diseases such as Alzheimer's. The lab's main research question is explaining how vascular dysfunction could lead to physiological as well as cognitive diseases such as Alzheimer's. My lab wants to answer our main question because Alzheimer's disease is a very large disease that affects a wide number of people, 2050 there was 41 million recorded cases of Alzheimer's disease, and it's projected in 2050 to have 131 million affected people. Well, this is important to try to answer because it is such a widespread disease. Finding a cure, or at least some way to lessen the ramifications of the disease, would be very beneficial. The big world issue that we're trying to solve is, can we delay or potentially even mitigate on the onset of Alzheimer's, especially at early onset Alzheimer's. So what I do is I stain slides with rat brains on it, and I add substances to it to make it glow, and then I go over to our confocal microscope and I take pictures of those slides, and then I analyze them later. The results of my experiment were that the addition of an antibody does not help to reduce plaque number or density. It actually increases the amount. One of the questions that I had originally was, what are the physics behind a microscope such as the confocal microscope that I did most of my imaging on? And a second question of mine is, what are the computer science aspects that would be related to a software like the ImageJ that we use?
Currently, I am working on a project with Dr. Josiane De Silva, and we are looking at the peripheral mesenteric arteries in 5xFAD Alzheimer's mice model. Currently, I am working on histology, so obtaining tissue specimens and staining them with H&E as well as picocyrus red to quantify the structural changes in 5xFAD Alzheimer's mice model. So the parameters that I had to explore include the lumen area of the peripheral vessels, as well as the vessel area, vessel wall area, and then comparing the lumen area with the vessel wall area to create a ratio, which will then allow us to analyze if the vessel has remodeling. And currently, there is a slight decrease, although it is non-significant, with 5xFAD lumen area, as well as a slight decrease in the vessel area. Furthermore, there is a slight decrease that is non-significant in the vessel wall area, and there is no change in the ratio, which is one of the parameters used to explore vascular remodeling. How can we improve obtaining tissue slices that are way too small and way too thin and easily collapsible? If there is no significant change in the remodeling index as well as the other parameters that we explored, what other explanation contributes to vascular remodeling and hypertension in Alzheimer's disease?
My lab's main research question is, can the drug known as PNA5 prevent inflammatory-driven neurodegenerative diseases? Dr. Pierre's lab and Dr. Hayes collaborate together to focus on whether the drug treatment of PNA5 improves antibody therapies for Alzheimer's disease. Dr. Hayes' lab wants to try to answer the main question to identify whether PNA5 prevents or reverses cognitive impairment and heart failure. The study is important. It could have the possibility to help people who suffer from heart failure and cognitive impairment. So in our lab, the big world issue that we're trying to solve is to reduce the high percentage of individuals who have heart failure with PNA5. Looking at preclinical animal models and clinical experiments on patients who have heart failure, and some experiments that are done are taking brain tissue samples and using an IgA1 marker to stain for microbial activation. So to contribute to the research, the things that I do in the lab are the process of cell counting and the skeletal analysis process. Being that my results are inconclusive, I learned from my past mistakes to try and improve on better future experiments.
Hello, my name is Nate Lichty, and I'm going into my junior year at University High School, and this is my SIMI lab video. I'm working in the Lichty lab, and my main lab mentor, Ken Lichty. The Lichty lab mainly focuses on wound healing and tissue regeneration. The main question of this lab is, how do we correct the wound healing process in diseases with impaired wound healing, such as diabetes? The main reason they want this answer to this question is to assist people with impaired wound healing, especially diabetics. To answer this question, the lab runs animal models like mainly mice and pigs with different examples like pressure ulcers, acute kidney injury, diabetic wound healing models, uterine wound healing models, and sepsis models. My role in this lab was to assist in the experiments by doing a lot of different stuff like RNA extraction, microscope analysis, cell culture, autoclaving, watching surgeries just so I can learn how to do it and then doing it myself, etc. Now let's begin the tour of the lab. Here we have the workbench where we're currently doing RNA extraction. Here is the centrifuge where samples are homogenized. This is the PCR test where we get the data for the RNA. That's the scale. And this is the homogenizer. This is the microscope where we analyze cells. This is where we warm up samples that have been kept in the fridge. That is where we make cell media and feed the cells. Here is the operation room where we will operate on mice or other animals and experiment on them to see their reaction to certain things. Overall, I've learned a lot about wound healing and experimentation and a lot of science in general. So, thank you for watching my SIMI video.
My name is Brody Stevens, and I'm a second-year student at New York University. I work in the Gertner Lab under the mentorship of Dr. Kellen Chen, and our lab focuses on wound healing and tissue regeneration. And our main research question in the lab is, what are the molecular mechanisms behind wound healing? Just to give you a little quick tour of our lab, this is some of our benches and working area. This is where we do a lot of our staining. And this is where we keep our hood, where we do fume and hood work. Today I want to share with you an exciting project we're working on to tackle a critical health issue, liver fibrosis. Liver fibrosis is a condition where excessive connective tissue builds up in the liver, often leading to cirrhosis and liver failure. This can result from chronic liver diseases such as hepatitis, alcohol abuse, and non-alcoholic fatty liver disease. Thiocetamide, carbon tetrachloride, and diethyl nitrosamine have all been explored for methods of inducing liver fibrosis, but the thiocetamide-induced liver fibrosis mimicked humic fibrosis most closely. Diethyl nitrosamine displayed liver cancer, and carbon tetrachloride was closer to replicating human liver fibrosis, but thiocetamide was deemed most similar to human fibrosis as it led to chronic liver injury and subsequent liver fibrosis. We inject the mice with thiocetamide for a certain duration of time to induce liver fibrosis, where we then let the fibrosis resolve, and then collect the tissue and liver and analyze it using serum analysis, histological analysis, MRI mapping, single-cell RNA sequencing, nanoindentations, and other various experiments to gather comprehensive data. This liver is from a C57 mouse that received 10 weeks of thrice-weekly TAA plus saline injections. Looking at some of the key features, in the first image we see blue staining, and in the second image we see purple staining. The presence of extensive blue and purple stained fibrous tissue indicates collagen deposition, a hallmark of fibrosis. The fibrotic bands are thick and widespread, suggesting advanced fibrosis. Most importantly, in both provided histological images, you can observe fibrous bands stained blue in the first image and purple in the second, extending from one portal tract to another, forming bridges. This is indicative of portal-to-portal bridging fibrosis. Working closely with liver specialist Dr. Belinda Sun at the University of Arizona and referencing the Ishak scale, which assesses the level of fibrosis, which scores range from 0, no fibrosis, to 6, cirrhosis, our model presents between a stage 3 and 4, but closer to 4. This is very promising, and more experiments can lead us to mimic a more cirrhotic human liver.
Although we've learned so much with this experiment, we still have so many remaining questions. Thank you.